Another vector designed specially for cloning larger fragments of DNA is called a cosmid. Cosmids behave both as plasmids and as phages. They contain the cos sites or cohesive sites of lambda phage DNA, which allow the DNA to be packaged into lambda phage heads, hence the cos part of the name cosmid. They also contain a plasmid origin of replication, so they can replicate as plasmids in bacteria, hence the mid part of the name or mid. So this is cos mid. So this is the diagram of cosmid. So you can see these green ones, these two are cohesive sites, cos sites. This is origin of replication, this is antibiotic resistant gene, and this is the site which contain restriction sites for various various restriction enzymes. Because almost the entire phage genome except for the cos sites have been removed from the cosmids, they have room for larger inserts up to 40 to 50 kb. Once these inserts are in place, the recombinant cosmids are packaged into phage particles in vitro. So this is the cloning procedure. So this is cosmid. It is cleaved at this polylinker site, so it will be linearized with a cos end. So after that the target DNAs are mixed with cleaved cosmids, and these target DNAs will be inserted here and then here and here. So in this way very large fragments can be inserted into this cosmid. These particles cannot replicate as phages because they have almost no phage DNA, but they are infectious. So they carry their recombinant DNA into the bacterial cells. Once inside the cell, the DNA recircularizes through its cos sites and from then on behaves exactly like a plasmid and replicate because it has a plasmid origin of replication. Cosmids are especially useful for the analysis of highly complex genomes and are an important part of various genome mapping projects. So another class of vectors that produce single stranded DNA has been developed. These are like the cosmids in that they have characteristics of both the phages and plasmids, thus they are called phagemids. One popular variety goes by the trade name P blue script or PBS. Like the PUC vectors, P blue script has a multiple cloning site inserted into the lac Z gene. So अगर lac Z gene है तो इसका मतलब है clones को हम blue white colony की basis पे select कर सकते हैं. This vector also has the origin of replication of the single stranded phage F1, which is related to M13. This means that a cell harboring a recombinant phagemid, if infected by an F1 helper phage that supplies the single stranded phage DNA replication machinery, will produce and package single stranded phagemid DNA. A final useful feature of this class of vectors is that the multiple cloning site is flanked by two different phage RNA polymerase promoters. For example, PBS has a T3 promoter on one side and a T7 promoter on the other side. This allows one to isolate the double-stranded recombinant phagemid DNA and transcribe it in vitro with either of the phage polymerases to produce pure RNA transcripts corresponding to the either strand of the insert. So, this is the phagemid. You can see the origin of replication, and here some selective markers are present, and here is the promoter of RNA promoters. So, this is a phagemid.